3Cells, and we're on part 3. You'll recall last time we were describing the peptidoglycan found in the cell walls of bacteria. So in this recording, we're going to look at 3 different types of bacterial cell walls: the gram-positive, gram-negative, and acid-fast cell wall. So we'll first focus on the gram stain and how it lets us distinguish between gram-positive bacteria and gram-negative bacteria. So the gram stain, which we will be doing in lab, is considered a differential cell in that it lets us differentiate between 2 big groups of bacteria based on their cell wall structure. So the 2 group of bacteria, one group, the gram-positive bacteria, will stain purple after we perform the gram stain, and the other group is going to stain pink after we perform the gram stain. And this difference in staining is related to differences in their cell wall structure. And it's a little bit hard to see over here, but these little pink guys here, these are gram-negative. And then these purple guys over here, these are gram-positive. So purple is positive, red or pink is going to be negative. And again, reflecting differences in cell wall. So these are nice cross-section through the 3 different types of bacteria cell wall. On your handouts, you'll have a little bit simplified version of these, but we'll use these to get us started. So here we see a cross-section through a gram-positive bacterial cell wall, and to get you oriented here, this would be the cell membrane, the cytoplasmic membrane. The cytoplasm is inside the cell, so the cell wall is going to be outside the cell membrane, this portion here. And what we see in gram-positive cell walls is a thick layer, multiple layers of peptidoglycan. So all of this purple represents a thick layer of peptidoglycan. And very importantly, peptidoglycan is very hydrophilic. Consequently, hydrophilic antibiotics have no problem diffusing through the cell wall. Hydrophilic stains, hydrophilic nutrients have no problem passing right through these thick layers of peptidoglycan of the gram-positive cell wall. Now, in addition, we'll see these little green filaments here. These are called lipoteichoic acids, and there's some controversy over their function. When I was in graduate school, there was great interest in whether these lipoteichoic acids played a role in letting gram-positive bacteria attach to or adhere to cells within a host. They usually have a net negative charge, so it might be that they do have some influence over what kind of molecules will be able to diffuse through the peptidoglycan. One thing I would like you to remember is that cells of our immune system have evolved receptors that can bind to lipoteichoic acid. Now, lipoteichoic acid will only be found in gram-positive bacteria. So cells of our immune system then have evolved receptors to identify this unique gram-positive bacterial lipoteichoic acid. And when cells of our immune system bind to the LTA, that will trigger an immune response. So we're always interested in unique molecules in any of our microbes. So do remember, lipoteichoic acid is going to be found in the gram-positive bacterial cell wall. Now, the gram-positive bacteria, when we gram stain them, right, the first step in the gram stain procedure is we're going to flood our cells with crystal violet, a beautiful purple color, and the crystal violet is going to bind to negative charges here in the cell wall, in the peptidoglycan. We are then going to add a sodium bicarbonate to increase the pH, and that will increase binding of our dye. And then we're going to add the mordant called Gram's iodine. Gram's iodine is going to complex with the crystal violet, forming these great big purple complexes. And then in the next step of the gram stain, we're going to add a decolorizer, alcohol acetone. The alcohol acetone, it's going to dehydrate our cell. It'll act as a lipid solvent, so it's going to dissolve this. And as it dehydrates our cells, these thick layers of peptidoglycan are going to collapse down around the cell, trapping the purple crystal violet iodine complexes within these multiple layers. So after decolorizing, our gram-positive bacteria are going to remain purple. That was why they were initially called positive, because they held on to crystal violet iodine. Okay, and then the last step in the gram stain reaction is we're going to flood our cells with a counter stain or secondary stain, this beautiful red colored safranin. But because the cell is chock full of this purple dye, and because the crystal violet iodine is binding to most of the negative charges, the safranin, there won't be sufficient safranin to kind of overwhelm the purple color. So our gram-positive cells will remain purple. Now, in contrast, let's take a look here at the gram-negative bacterial cell wall. And again, to get you oriented inside the cell, the cytoplasm, this is a cell membrane. The cell membrane is not part of the cell wall. And we see in the gram-negative cell wall, we have only a thin layer of peptidoglycan right here. And then we have this unique outer membrane. Now, the outer membrane, folks, is not a cell membrane. The outer membrane is part of the gram-negative cell wall, and the inner leaflet here is going to be rich in phospholipids, and in this outer leaflet we will see some phospholipids, but there will be a second toxic molecule in the outer membrane, and this is called lipopolysaccharide LPS, and we're going to see this is toxic. When gram-negative bacteria are growing in our bodies and they start to die, they release this lipopolysaccharide. It's also known as endotoxin, and that's going to trigger an exaggerated inflammatory response, which can actually kill us. It can lead to sepsis, septic shock, and we can actually die from this. So we'll come back and talk more about that. Lipopolysaccharide is unique to gram-negative bacterial cell walls, and luckily our cells of the immune system have also have evolved receptors to bind to lipopolysaccharide. So again, binding of the lipopolysaccharide to cells of the immune response of the cells of the immune system will trigger a strong immune response. All right, so this outer membrane, we could say between the phospholipids and the lipopolysaccharides, this portion is lipid. We could say this is a lipid bilayer, and thus it's going to be hydrophobic. So it would prevent diffusion of large hydrophilic molecules across the outer membrane. So what does the cell do to transport hydrophilic molecules across the cell membrane? Well, they'll have proteins in the outer membrane, and these proteins, some of them will act as porins. There'll be proteins with a water-filled channel through which large hydrophilic molecules can cross. Now, one thing we want to point out right here is that the first antibiotic discovered, penicillin, is a large hydrophilic molecule, and in most gram-negative bacteria, penicillin can't cross through the outer membrane porins. And as a result, this means most gram-negative bacteria are naturally resistant to penicillin because the penicillin can't cross the outer membrane. It can't get down here where the transpeptidase are working. So again, most gram-negative bacteria are naturally resistant to penicillin. The solution was chemists took penicillin and they chemically modified it to make what we call semi-synthetic penicillins or semi-synthetic beta-lactams such as ampicillin and amoxicillin. And the beauty of ampicillin and amoxicillin is that those two antibiotics can cross through the outer membrane porins. So thus, we say that ampicillin and amoxicillin can be used to treat gram-positive bacteria and also gram-negative bacteria. We'd say ampicillin and amoxicillin have broad spectrum, can be used against both gram-positive and gram-negative bacteria. In contrast, we say usually penicillin is narrow spectrum, as it's usually most useful against gram-positive bacteria. Usually you wouldn't choose penicillin as your first antibiotic to treat a gram-negative bacterial infection. Okay, what else do we have here? This little space between the outer membrane and the cell membrane here, this is called the periplasmic space. It's where we'll find our peptidoglycan, our transpeptidases. There'll be enzymes in here. And the last little bit, I don't ask this on the exam, you guys, but this little connector between the peptidoglycan and the outer membrane, that's called lipoprotein. But again, I won't ask that on our first lecture exam. The third type of bacterial cell wall is the acid-fast cell wall. And luckily, only a few pathogens have this waxy acid-fast cell wall. The wax we usually describe is called mycolic acid. It has an incredibly incredibly long hydrocarbon tail, so a nonpolar hydrophobic tail. And this outer layer, this waxy layer, then creates a hydrophobic barrier, protective barrier, almost like a raincoat, right? So it's going to inhibit diffusion of water-soluble antibiotics, water-soluble dyes, stains, will inhibit diffusion of water-soluble hydrophilic antiseptics and disinfectants. So who amongst our bacterial pathogens have these waxy cell walls? The genus called Mycobacterium, and there's two important members, two species of Mycobacterium that are significant pathogens: Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes Hansen's disease. Now, the consequences of this waxy um, cell wall is that you have to choose your antibiotics very carefully. Your patient has to be on antibiotics to treat, say, Hansen's disease or tuberculosis for months, maybe years, because the antibiotics diffuse slowly through the cell wall. The acid-fast cell wall is really hard to stain. If we tried to gram stain it, it probably wouldn't stain. And that's why a special staining procedure, the acid-fast stain, was developed so that we could stain these little guys. And we'll talk about the acid-fast stain in lab. The waxy wall prevents the bacteria from drying in the environment. So if I had active TB and I'm coughing out Mycobacterium tuberculosis into, say, my room, those bacteria, they wouldn't dry up and they would remain infectious for long periods of time. The waxy wall makes these bacteria slow growing, and that slows down diagnosis. It slows down antibiotic sensitivity testing. And furthermore, this waxy wall makes it really, really hard for cells of our immune system, our phagocytes, to destroy the acid-fast bacteria. So an abbreviation you'll see in describing acid-fast bacteria is AFBs, AFB, acid-fast bacteria. And our two examples again are Mycobacterium tuberculosis and Mycobacterium leprae. All right. I just realized that I forgot to describe how our gram-negative bacteria will react in the gram stain. So let me just back up. So folks, you'll remember that in our gram-positive bacteria, this thick layer of hydrophilic peptidoglycan, when we do the gram stain, the crystal violet iodine, the purple color, will get trapped in these layers of peptidoglycan when we decolorize, and thus gram-positive bacteria will appear purple after the gram stain. So let's come down here though and look at what's going to happen to our gram-negative bacteria cell walls. So recall, folks, that the gram-negative bacteria, they just have this thin layer of peptidoglycan. When we gram stain them, the crystal violet and iodine will form a complex, and it's believed, a recent paper suggested that the crystal violet iodine is binding mostly to the peptidoglycan here. Okay, so initially the gram-negative bacteria will stain purple with the crystal violet iodine. But when we come to the decolorization step, when we add our alcohol and acetone, the outer membrane will be dissolved, and remember, the cell membrane will be dissolved. The cell will dehydrate, and this thin layer of peptidoglycan will collapse down around the cell, but because it's so thin, it can't trap the crystal violet iodine complexes. So when we decolorize, the crystal violet iodine complexes are going to get washed out. So the gram-negative bacteria become colorless. We say they're decolorized. Now, colorless bacteria, they're hard to see. So we add a secondary stain, a counter stain, in contrasting color called safranin. So we're going to flood our gram-negative bacteria after they're decolorized with safranin. The safranin will bind to negative charges here, and thus the gram-negative bacteria after gram staining will appear red or pink. Okay, so remember, in gram stain, gram-positive bacteria like the genus Staphylococcus, the genus Bacillus, the genus Streptococcus will appear purple, and gram-negative bacteria such as E. coli and Salmonella, Vibrio cholera will appear pink. One thing we'll talk about in lab is when we do gram staining, we want our gram-positive bacteria to be young, less than 24 hours of age. And the reason is, if they aren't young, their peptidoglycan starts to break down. There's enzymes that will actually start causing holes to form in the peptidoglycan. So if we gram stain old gram-positive bacteria, When we decolorize them, there's so many holes in the peptidoglycan, we wash out the crystal violet iodine, they decolorize, and then when we counterstain with safranin, these old gram-positive cells will appear pink or red. So we think they're gram-negative, but they aren't, and that's called a false gram-negative reaction. That's something we have to be really careful about in the lab. Okay. So this is just from your handout, folks. So just, again, really quickly, the three types of bacteria cell walls. Just for orientation, this is the cell membrane, cytoplasmic membrane. Cytoplasm is here. This is a gram-positive cell wall, thick layer of peptidoglycan with lipoteichoic acids, teichoic acids. The gram-positives will stain purple with the gram stain. Here's our more delicate little gram-negative bacterial cell wall, thin layer of peptidoglycan. Here's that cool outer membrane, a lipid bilayer, hydrophobic barrier. We have our phospholipids here, and here's our lipopolysaccharides, our toxic molecules, our endotoxins. And here's a lovely little outer membrane porin. And remember, large hydrophilic substances have to have the right size and shape and charge to pass through the porins if the substances are going to reach the periplasmic space and then subsequently the cell membrane. Here's our acid-fast bacterial cell wall, as we would find in Mycobacterium. The acid-fast bacterium are probably more closely related to gram-positives. They have a moderate layer of peptidoglycan right here, and here is then that waxy outer layer, rich in mycolic acid. That's the wax I'd like you to remember. So the mycolic acid here going to be found in our acid-fast bacteria. Notice, you guys, that mycolic acid is not in the gram-negative cell wall nor in the gram-positive cell wall. And just a little bit more, folks, on the lipopolysaccharide of the gram-negative bacterial cell wall, the outer membrane. So it's often abbreviated LPS. The P stands for polysaccharide, and this is detail we won't need to know right now. It's often referred to as the O, or somatic antigen. We can mount an immune response against the different types of polysaccharides in the different lipopolysaccharides. It's the lipid portion, and the lipid portion is called lipid A. This is the true toxic portion. So on an exam, folks, if I asked you what is endotoxin, you could tell me either it's lipid A or you could say it's lipopolysaccharide. Where is it found? It's found in the outer membrane of gram-negative cell walls. So when our gram-negative bacteria die, if they're growing in our patient, say in the patient's bloodstream, when the bacteria die, they start falling apart and they'll release massive amounts of lipopolysaccharide, which then bind to cells of the immune system. And that triggers cells of our immune system to release huge amounts of these chemical messengers we're going to call cytokines. Now, normally, cells of the immune system will release little amounts of cytokines just locally. The cytokines are used as chemical messengers between cells of the immune system. But if we have lots of gram-negative bacteria dying, we get massive cytokine release. They call it a cytokine storm. And as a result, we get inappropriate responses. We get vasodilation, our blood vessels get bigger, they get leaky, we have increased capillary permeability. Vasodilation in leaky vessels means the blood pressure will go down, and that's going to decrease blood delivery to our organs. Another consequence of endotoxin release is that it triggers the clotting cascade. So now we're going to start throwing little blood clots everywhere. And that's a phenomenon called DIC, disseminated intravascular coagulation, that further blocks blood flow to our organs. We end up with multiple organ system failure, right? Hypotensive shock, and this can lead to death. So this is often called endotoxic shock or gram-negative sepsis, and it can happen very rapidly. So this is kind of a nightmare in emergency rooms or in hospitals, recognizing if your patient is in the early stages of endotoxic shock. So just with regard to therapy, often what you try to do is give lots of IV fluids, trying to get the blood pressure back up. You might have to give blood transfusions, right, because all the clotting factors are getting used up. If your patient uses up all their clotting factors, ironically, then they'll hemorrhage, right, because there's no more clotting factors, so often you need to give them blood. Because you could have damage to the kidneys, you might need kidney dialysis, you might need ventilatory support, and it's really sad because often folks who recover from endotoxic shock, sometimes they have long-term sequelae like kidney damage, and sometimes there's some damage to the brain, so there can be mental changes afterwards. So this is a tough one. I think we're going to stop here. No, we're not going to stop here. So why do we care about the different types of bacterial cell walls? Really important, folks. Like, it's like one of the first steps in trying to identify an unknown bacterium, to know if it's a gram-positive bacterium, gram-negative bacterium, or acid-fast bacterium. Really important. And then knowing the identity of the pathogen is going to help with determining maybe prognosis for the patient. It also give it an early indication, possibly what kind of antibiotics you should use. And that's another reason we want to identify the type of cell wall, because remember, gram-negative bacterial cell walls usually won't let penicillin cross. So you don't want to use penicillin on gram-negative bacterial pathogens unless you identified them ahead of time. The acid-fast bacterial cell walls, many antibiotics cannot cross it. So again, knowing the type of cell wall is going to be a huge factor in choosing the correct antibiotics for your patient. And I think we're almost finished. So some bacteria lack peptidoglycan, and the one group I'd like you to remember is the group of bacteria called mycoplasma lack peptidoglycan. They don't have cell walls at all. And the reason that's important is that if you had a patient with a mycoplasma infection, maybe a mycoplasma infection of the lungs, so-called walking pneumonia, you wouldn't choose a beta-lactam antibiotic, because the mycoplasma don't have the target mycoplasma don't have the transpeptidase, they don't make peptidoglycan, so a beta-lactam antibiotic wouldn't be useful. In the next part of these audio recordings, we will talk about other targets within bacteria. For example, the 70S ribosomes, and we have antibiotics that will target the bacterial 70S ribosome. So, for example, we could use one of those to treat a mycoplasma infection. We do want to remember only members of domain bacteria make peptidoglycan, so members of domain archaea don't. If we stained archaea, some of them might appear gram-positive, but it's because they have thick walls of proteins or carbohydrates. It's not peptidoglycan. And again, some archaea might stain red in the gram stain, gram-negative. But again, they don't have any peptidoglycan. Thin layer of protein replaces their outer membrane. Now, this is a really important question, you guys. If we identified an archaea human pathogen, let's say there was an archaea that caused infections in humans, would beta-lactam antibiotics kill them? So the thought process here, you guys, you have to know that beta-lactams are going to target the transpeptidase that cross-links peptidoglycan. So you identify the target of the antibiotic. Then you ask, does your pathogen have that target? So we'd ask, do archaea have peptidoglycan? And we'd say no. Therefore, we know that beta-lactams won't kill archaea, right? They lack the target of the antibiotics. So with this, folks, we are going to close this part 3, bacterial cell walls. And in the last part of the video, we'll go over the other layers of the bacteria, the glycocalyx. We'll talk about fimbriae, sexpili, and flagella. And then we'll go inside the bacterium to talk about the chromosome, to talk about plasmids, the 70S ribosomes, and endospores.